I'm going to actually switch gear to back to the taste system where Bob made his original really landmark discovery for gustation, mark the molecular biology of taste. So today I'm actually going to talk about taste stem cells. So the sense of taste, humans and many other species can perceive 5 basic taste qualities, include sweet, umami, bitter, sour, and salty. As you can see from these pictures, they all have a lot to do with what we eat. So the sense of taste really influences our dietary intake. So detection of taste starts in the oral cavity. So these taste buds are teeny sensory organs responsible for detecting taste stimuli. So where you can see here, anterior tongue, you have this so-called fusiform papillae. In the back of tongue, you have so-called 48 circumvallate papillae. Also, not just on the tongue surface, in the soft palate, so the upper cleft region, there are also taste bud cells you can find. Even more so in the actually oral regional nerve, you actually can find taste buds as well. So taste buds actually scatter across many different taste fields in the oral cavity. When you having a closer look up of a mouse tongue tip, where you can see here this rod whitish structure, those actually are fusiform papillae. When you zoom in again, you can see this taste bud containing like about 50 to 100 cells. Here, you already heard this morning. So this is actually the tryptophan-5 GAP, so the green cells, those representing sweet, bitter, umami cells. Although here I show you a really static image, in reality, actually taste bud cell turn over frequently throughout life. So the average lifespan of a taste cell about like 10 days. So this senescent dead cell continually slow off from taste buds, and new cell generated by adult taste stem cell to replenish taste buds. This process actually depends on innervation. So a phenomenon observed many years ago, actually 150 years ago. So when I tried to read this paper, this original discovery of dependence of innervation, dependence of taste tissue homeostasis on innervation. So this is actually from 2 German scientists. I could not really understand German, but with the Google Translator, I actually be able to read it. I was fascinated because originally this paper really tried to identify which nerve innervate taste bud in circumvallate papillae in rabbits. So what they found is once you cut nerve, this taste bud degenerate. So later study from many groups show taste buds reappear once the nerve grew back. So based on all this evidence, here's a conceptual model we actually have for taste tissue homeostasis. So taste bud cell again turn over, and the stem cell need to replenish the system, and that rely on gustatory nerve. You will see this model actually throughout my talk. So my lab have been interested in really know how this works for the past decade. So first, I'm going to show you today is how we identify taste stem cell. Then I'm going to show you how we actually using stem cell in dish to actually grow this structure called taste organoid. So that recapitulate what's happening in native tissue. Finally, I'm going to show you molecular and cellular mechanism for neuronal regulation of taste stem cells. And when we start really looking for markers to mark taste stem cell, at that time, it was reported by our keynote speaker, Dr. Hans Clevers, that LGR5 marks stem cell in the intestine. So because we found LGR5 also expressing taste tissue, so we asked whether or not LGR5 expressing cell also mark adult taste stem cell. So you can see from this picture, this is a green fluorescence. So this is a genetic mouse model generated in Hans Clever's lab, and this under the control of LGR5. So this knock-in mouse model expressing GFP at the Cre-ERT. So GFP for tracking this cell, Cre-ERT, so you can actually do the linear tracing using this. So where you can see these cells actually surrounding or at the base of T cell. So appear to be at the right place to do the right job. Can they do that? So we use this linear tracing technique. So that's very standard for studying cell fate determination for determining the regenerative capacity of a stem cell. So again, this is the mouse line, LGR5 driven the expression of Cre-ERT. You can cross with the reporter line, and this reporter line expresses TD tomato, but only when this Cre-ERT be activated by tamoxifen injection. So this basically, this is pretty straightforward. So we can turn this green cell into red. Any daughter cell derived from this green cell will become red. So if they represent stem progenitor cells. So here's what we see. So immediately after tamoxifen injection, you can see this green cell turn to red. But over time, you can see there are many more red cells, including taste bud cell and cells surrounding taste bud in the circumvallate papilla, suggesting this LGR5 has the capacity to generate both taste and non-taste cell in circumvallate papilla taste tissue. So next, we actually try to ask a question of what exactly the regenerative capacity of this cell, because we know taste buds contain 50 to 100 cells. This actually, there are many different types of taste cell in taste bud. We ask whether or not a single LGR5 stem cell be able to generate all different types of taste cells. So that's actually really the question in stem cell biology. So for that, how you answer that question, we actually developed this so-called taste organoid system. So again, this is from pioneer work by Hans Cleaver. He's pioneered the organoid technology. So we ask whether or not we can do this in taste tissue using taste tissue. But remember, I told you before, taste tissue homeostasis require innovation. How you do this without neuron? So what we speculate in our culture medium, perhaps that already contain factors secreted by neuron. So where you see actually a talented postdoc in my lab at Wen Zheng, she actually can dissociate this LGR5 GFP cell into single cell, that sort this LGR5 cell out, that grew them in dish, where you can see this single cell. Of course, you cannot really see it, single cell, but over time, you can see it grow bigger, they actually containing T cells. And by day 7 or 9, you actually can see by your naked eyes when you grab this dish and take a look, they actually there. So quite remarkable. So here is a movie. Actually, this is not from LGR5, isolated, sorted LGR5 cell, but from other mouse models. Because later on we realized, as long as you have stem cell, they can grow. Doesn't matter, you need to sort them out or purify them, but they can do that. So this is actually, again, from this 3.5 GFP mouse model. Let me see if I can play this video. Oh, anyhow, I'm not going to play this video. Okay, thank you. So I really want to show this video because it's so cool. So what you can see here is these green cells. So this is 3.5 GIP you already heard. So those are sweet, bitter, umami cells. Also co-stay with the antibody against gasteusin in this case. So those are sweet, bitter, umami cells. Segregated from this population, you also see these white cells. Those are actually sour cells. So exactly like what happened in native tissue. So single adult stem cell actually can give rise all different types of T cell in dish. So next we ask whether or not these cells are actually functional. Do they respond to taste stimuli like sugar, so on, artificial sweetener, bitter taste compound? You heard taste receptor, the taste signal cascade, there are calcium mobilization involved. So we ask whether or not we can just use calcium imaging to see whether or not taste receptor cell in organoid can respond to taste stimuli. So here I show you this is a sweet taste cell in organoid respond to artificial sweetener like AC-SUMK and the sucralose. And here's another cell respond to denatonium. You heard denatonium in mice, actually. In human, denatonium intensely bitter to us, but not so much to mouse. So that's why you need to use quite some denatonium there. Also, this response can be inhibited by a PLC beta 2 blocker U73122. And once you wash off this blocker, the cell regain responses to denatonium, suggesting this is a beta receptor cell. Basically, by morphology, by transcriptome analysis, by functional, they exactly like taste cell. So we build a culture system that can recapitulate what's happening in native tissue in terms of taste tissue homeostasis and the taste receptor function. So back to this question, neuronal regulation of taste stem cell. So we show like taste stem cell can grow, generate a mature taste cell when cultured in dish in the absent innervation. So what exactly is gustatory nerve derived factor regulate taste tissue homeostasis? How we study this? So for that, I just illustrate the concept again. When you cut nerve, so this nerve trisection model, so taste bud disappear. The reason for that is because stem cell become quiescent, so not active. So that's what happens. When you already have stem cell, there's no taste bud cell anymore. So what if we actually put this putative factor back into the system? We should be able to promote taste cell generation. So this is exactly what we did. First, we built this nerve trisection model, where you see here is a sham-operated mouse model. You can see taste bud cells, and they are heavily innervated. This is actually sour tasting cell. When we actually cut this nerve, you see this taste field no longer innervated. There are also some only residual immunoreacted in this mesenchyme core. There are also no taste bud cells really left, only residual ones. So using this model, we actually try to provide this putative niche factor back into the system. So what exactly the factor we should work on? So because I mentioned LGR5 marks taste stem cells, so we ask whether or not the ligand for LGR5 responding actually is the niche factor produced by neuron. There are 4 respondings. They actually all can do the same thing, binding to either LGR5 or ZRF RNF E3 ligase. So this is another pair of receptor I'm going to discuss later on, if I still have time. So what you can see is one of these are responding actually expressing gustatory neurons, including geniculate ganglia, nodose petrosa ganglia, but not in circumvallate papillae. So suggests this is derived from neurons. And then we ask if we can, if we provide this responding back into the system in this nerve trisection model, what happened? So for that, we use this adenovirus-based approach, actually pioneered by Kevin Ku's lab. So his lab happened to have this adenovirus encoding RSpawn1. So we ask whether or not you actually infect mice with RSpawn1 using adenovirus. So they actually infect liver cell, produce a lot of RSpawn1 in the system, in circulation. Can they actually promote generation of taste cell in the absence of innervation? So here's what's happening. You can see that there's a lack of innervation, taste nerve cut, but there's numerous taste bud cells in the circumvallate papillae. If you look at the control, basically there's a control adenovirus encoding only the FC fragment, nothing really happened, suggesting the responding can promote taste cell generation. And I also want to highlight this part. Actually, typically you don't really see many taste cells in this region. The reason why we think we see these because we feel like this circulation, responding circulate in your system. So wherever they see a stem cell or even quiescent, or maybe they actually can, because there are lots of responding, they can promote this stem cell population expansion. So wherever, so that's why you can see many more taste buds taste bud and the taste bud cells. Now those like SAR cells, where we actually using another marker to mark the sweet bitter umami cell, you can again see numerous taste bud in the circumflexion papillae. In contrast, when you see the control, there's nothing really happening. The taste buds all disappear. Again, in this dorsum region, you can see ectopic taste buds. Not just R-spondin 1, because I mentioned R-spondin 2 produced by gustatory neuron. So we also use the adenovirus approach combining with this nerve trisection model. We can see R-spondin 2 can do the same thing, where you can see the sour tasting cell, non-innervation, but you actually have many cells when with the mice receiving adenovirus encoding R-spondin 2, but nothing with the control. So when we actually, this demonstrates the sufficiency of R-spondin to do the job, but whether or not R-spondin is required, we don't know. So for that, we first test this in organoid culture with or without R-spondin, where you can see is, should I stop?
Yeah, you have 2 minutes, but no questions.
Oh, okay. I'll be fast, but I want to show you actually, maybe I skip that slides. I'll show you here is whether or not R-spondin is really required. And what we see here is we try to conditionally ablate R-spondin 2 from the system. But this is actually R-spondin 2, the gene structure is very unique because each one, they have 6 exons. They all actually in frame with each other. There's 2 important exons, one encoding the binding domain for RNA 4303, and the other is encoding LGR5 456. So what we try to do is actually remove all these critical domain from system. That's actually very hard, very easy to say, but very hard to do. Luckily, actually, when we all joined Monell, we actually got a really new technique to use. Basically, we can actually deliver Cree into the nodose jugular ganglia complex. For that, we can actually ablate our response too. So what you can see is when we infect mice, inject the virus into wild-type mice, nothing really happens. You can see this green cell at the innervated. But in the once you ablate R-spot2, you see there's no taste buds, and also this nerve terminal retract, so the innervation not really in the taste field either. So clearly, this demonstrates the necessity of this R-spot2. Because of time, I'll just actually finally show you some of the thing I think it's pretty striking. So R-spot2 bind to like LGR5, also bind to ZRF RNF. ZRF RNF are E3 ligase, so the purpose of them is to stop the stem cell from working. So R-spondin is just stop that. So R-spondin is really a mass switch. So shown here is actually we do this whole mouth staining of tongue, where you can see this fusiform papillae containing 1 taste bud. At the circumvallate papillae, foliate papillae, there are also some taste bud in this area called epiglottis. So this is actually after a long time ablation of this receptor. You can see numerous taste bud. I don't know how many times, maybe hundreds. In areas such as this area is fully covered with taste bud. It's not just on the tongue, but also on this upper cleft. Wild type, typically you see this V-shaped taste buds, but when you actually ablate RNF at the ZRF, this is what happens. You can see numerous taste buds. I hope today's talk I can convince you this mechanistically how this works. Actually, neurons really secrete R-spot2, serve as a mass switch of taste cell renewal and the taste bud maintenance by suppressing RNF and the ZRF activity in taste stem cell progenitor cell by regulating one signaling in both in vivo and in vitro. With that, I'll stop my talk, acknowledge people in my lab and also collaborators, including Bob. Thank you so much, Bob, for your mentoring and for your support over years. Also, my work funded by NIDCD and other organization. And I also especially thank Kevin Koo for providing us with the adenovirus vector, and Hans Clevers and BK Koo for providing us with the ZRF RNF flux mice. With that, I'll be happy to answer any questions. Sorry, I got a little bit long.